Good day, everyone. This video is talk about the liver structure. I'm Mr. Ong from College Tingkatan 6 Seliputra. Previous lessons, we have discussed about the physiological and behavioral control in thermal regulations of endotherms. Today, we would like to describe the structure of liver and explain the role of each component. First of all, let me see the anatomy of the liver. Liver is the largest internal organ in the body and divided into two main blocks. Here you can see the first one, the larger, and the smaller one. And the liver contains up to 100,000 polygonal blocks of cells that call lobules. Okay, so when we cut the liver structure, we can see the liver they consist of 100,000 polygonal blocks of cells. Okay, we call them lobules. These are known as liver lobules. And then between the lobules are branches of the hepatic artery, hepatic portal vein, and the bile duct. So this is one lobule. This is another lobule. So they say between the lobules, you can see they are the branches of hepatic artery, okay, the hepatic portal vein, and the bile duct. And then at the center of each lobules is they have a central vein. Okay, we call it as a central vein, which carry the blood, okay, from livers to the vein, and then returns to the heart through the vena cava. Okay, so this is the lobule structure. One lobule, you can see at the center they have a central vein. Okay, and then between or at the edges of each lobule, so you can see they have three branches. Okay, so one is the hepatic artery, then another one is the hepatic portal vein, and then you also have the bile duct as well. Okay, lobules are formed from radiating rows of vertical cords of hepatocytes, which are the liver cells, radiating out from the center like spoke in a wheel. So from here you can see when we enlarge this a part of the lobule. So this is a central vein, and then you can see that they have they're formed by many of these hepatocytes, okay, or liver cells that radiating, okay, that radiating out from the central here. Okay, so they form a lobule. Then you see here they have three what we call the branches. Okay, one is called the bile duct. Then the other one is the hepatic portal vein, okay, which carry the blood from the small intestine, and also the branch of the hepatic artery that carry the oxygenated blood from heart. Okay. Then this is the prepared slide, okay, of a real liver lobules. So first of all, you want to identify the lobule, the liver lobule. First of all, you must identify the location of central vein, okay? Because every lobules, okay, every liver lobules, they have one central vein, okay? So then you can see the hepatocytes. The hepatocytes, they are radiated out from the central vein here. Okay, and then this is one of the edge of the lobules. So here there are three branches. Okay, once you can see which has the largest, okay, lumen is known as the hepatic portal vein. Okay, and then the other one is they have the smallest lumen. Okay, and then rough inner surface. So this is known as the hepatic artery. Okay, if there was smaller ones, we can call it as a hepatic arteriole. Okay, and then the bile duct, okay, which is here. So this is a lymphatic vessel. We are not consider this one in our syllabus, but at least you know. Okay, so maybe in order to distinguish between this hepatic arteriole and the bile duct, okay, because they also small. These two are smaller vessel or smaller duct. So from here we can see in the hepatic artery they are the smooth muscle, smooth muscles that they found in the hepatic arteriole. And also sometimes you can locate the red blood cell or the blood cell inside this lumen. Okay, and then you compare to the bile duct. Okay, so the liver actually is supplied with blood by two blood vessels. One is the hepatic artery, and the other one is the hepatic portal vein. So the hepatic artery leads from the aorta and delivers oxygenated blood, okay, from heart to the liver. Whereas the hepatic portal vein that leads from the small intestine and delivers the blood rich in absorbed nutrients, okay, especially the one water soluble nutrients from the small intestines to liver. So this is the schematic here for the blood flow. So the blood is the arterial. So when they goes to the stomach, okay, and then they go to the intestine, small intestine. So the nutrients are the water-soluble nutrients, glucose, amino acid, and the water-soluble vitamins. Okay, vitamin B complex, C, they are absorbed into the blood capillary, and then will channel to the hepatic portal vein to the liver, okay, to liver. So in the liver later, this blood, okay, from the hepatic portal veins, so they will channel to the central vein, and then which is transported to the venous blood and then to the vena cava and returns to the heart. Okay, so this is a hepatic vein, and after that the central. These are central hepatic vein. That means from the central vein first, then go to the hepatic vein. Okay, so this is the hepatic portal system. Okay, then the other ones from the hepatic artery, so which carry the blood from heart, okay, to the liver, okay, to the liver, and then also channel to the central vein and then lead to the hepatic vein. Okay, then the hepatic veins in the center of each lobule carry blood away from the liver, okay, to the vena cava and returns to the heart. Okay, so this is what we have discussed just now. So this is from this way, the artery. The artery. So here see the liver they accept or receive the blood, okay, from two destinations. One is through the hepatic artery, then the other is from the small intestine through the hepatic portal vein. Then after that, the liver and the blood in the liver will be channeled out, okay, or transported out of the liver through the hepatic vein, okay, and then to the vena cava and returns to the heart. Okay, these blood vessels they join to form sinusoid. So in lobule, like just now, we see the hepatic artery, and then they are the hepatic portal veins. So they will channel the blood, okay, to the central vein. Okay, then before they enter the central vein, you can see the blood from the hepatic artery and the blood from the hepatic portal vein, they will mix. Okay, so they mix in a channel of that surrounded by hepatocytes. Okay, so this channel is called sinusoid. Okay, sinusoid. So from here you can see a lot. Okay, then the blood flow from the sinusoid through the lobule, okay, and into the branch of the hepatic vein. Okay, at the center of each lobule. Okay, so the hepatocytes, okay, hepatocytes are structurally undifferentiated and all are identical. Then the cytoplasm contains mitochondria, okay, because the metabolic rate in the liver cell or the hepatocytes are higher. So therefore, they have a lot of mitochondria to supply energy. okay, for the biochemical reactions. And also Golgi apparatus, because they carry out metabolism, and then they also produce what we call a lot of enzymes and plasma protein as well. Lysosome. Okay, one of the function of the liver cell is to detoxify. Therefore, they have lysosome. And then liver cells, they store large amount of glycogen. Okay, therefore they have a lot of glycogen granules. Okay, and the fat droplet as well. Okay, the hepatocyte play the vital roles in metabolism, detoxification, and bile production. Like just now we can see they have a lot of those organelles. Okay, they involve in metabolisms, detoxification, okay, and the bile productions. Okay, the metabolism that involve the carbohydrate metabolism, protein metabolisms, and fat metabolism. The surfaces of the hepatocytes facing the blood, okay, they mean facing the sinusoid, okay, they have microvilli, okay. So here's a sinusoid. So this is the hepatocyte. So the surface, the plasma membrane, and the membrane surface of the hepatocyte that faces the sinusoid, they have microvilli. When you see the microvilli, that means they will increase the total surface area for reabsorption, okay, or for secretion. So this one we call it as materials exchange. Okay, from the liver lobule, now we go to the smaller or functional, what we call the structure, which is known as the liver acinus. Okay, plural form is acini. It's the smallest functional unit of the liver. Okay, so it includes two triangular portion, okay, of two adjacent lobules that is served by a hepatic arteriole, okay, crossing the center. So here's one lobule, here is the other adjacent lobule or the neighboring lobule. Then you can see the hepatic artery, okay, at the center that supplies the blood, okay, across the center. Okay, actually it's the edges and the barrier here lah. Okay, with the central vein at two opposite corner. Okay, the central veins at the two opposite corner. So this structure, okay, that consists of two triangular portion of the two neighboring nodules. This structure is known as liver acinus, okay, or the liver acinus. Okay, normally in when we study, we just show a part of an acinus. Okay, so the part of acinus that's showing here is from a central veins, okay, to what we call a branch of the hepatic portal vein and the hepatic artery and the bile duct, okay, to the other end. Okay, so from this structure, you can see the channels which carry blood between these rows of the liver cells are called sinusoid, as we discussed just now. So the sinusoids are open channels, okay, through which blood flow, okay, from the hepatic portal vein and the hepatic artery, okay, and then they have the direct contact for the hepatocytes, okay, for material exchange, and towards the central vein. So that means the blood from the hepatic portal vein, which is rich in nutrients, okay, from the small intestine, they will mix, okay, with the oxygenated blood from the hepatic artery, okay, in the sinusoid, okay, then before they channel into the central vein. The blood flows into the liver from the hepatic artery and the hepatic portal vein is mixed as it flows into the sinusoid. So from here, remember, into the sinusoid. Okay. Then the sinusoids are lined by numerous phagocytic cells, okay, which are known as Kupffer cells. So that means Kupffer cells, they are phagocytes, okay, white blood cell. They are involved in the breakdown of the old red blood cells and the injections of potentially harmful microorganisms that enter the blood circulation, okay, especially from the small intestine, the through the hepatic portal vein. Okay, this is the Kappa cells. Other channels carry bile are called bile canaliculi. Okay, so this is bile, okay, the bile duct. Okay, so the bile actually is produced by the hepatocyte. Okay, so this hepatocyte, they were produced bile and released into the bile canaliculus. Okay, singular form is a bile canaliculus. Okay, and then after that, they channel to the bile duct, and then from the bile duct, they will channel the bile into the gallbladder, okay, to be stored. Okay, the role of the liver has a very large number of functions, all of which contributes directly to homeostasis and can be conveniently divided into 4 categories, okay, according to our syllabus. So first is the bioproduction, okay, bioproduction, like just now we already discussed, and storage of vitamins. Certain vitamins can be stored in the liver, and certain minerals as well, and also the storage of blood, because the liver is the largest visceral organ, so they store a lot. It acts as a blood reservoir, okay. And detoxifications, okay, also carried out in the liver, and hormone breakdown, and metabolite conversions. So this metabolite conversion, the metabolites are referring to the carbohydrates, proteins, and fats. Then other role of the liver is heat production, because we say the liver, they act as a blood reservoir, and then they also have a high rate of metabolisms. So catabolism, especially, they produce heat, right? And formation of the red blood cell in the fetus, when during the fetus, the red blood cells are produced in the liver cell, in the liver, not liver cells in the liver, and also the breakdown of hemoglobin and iron storage in adults, okay, or after birth, after the baby delivery. All right, so from here you see production of bile. Bile is a yellow-green alkaline mucus fluid containing the bile salt and the bile pigments. So the bile salt, okay, sodium glycol and the sodium taurocorate, which are synthesized from cholesterol in hepatocytes. So that means the bile salts are derived from cholesterol, okay, in hepatocytes. Then the bile pigments are derived from heme residue. Okay, bile salts are secreted into bile along with cholesterol and phospholipid in the form of the large spherical particles called micelles. Okay, like this one. Okay, like here. So the function of the bile salt actually is for emulsification of fat, okay, in the duodenum and ileum during fat digestions. Okay. Then at the same time, the bile salt also activate the enzyme, okay, lipase. So this is a fat globule. So with the bile salt, so they will emulsify the fat globule into the tiny fat droplets, and then this tiny fat droplet will be digested by lipase easily. The bile pigments have no function actually, and to be removed. Okay, so the worn-out red blood cells are engulfed by the Kappa cell, and their hemoglobin is broken down. Then they'll store the ions and the heme residues. They will be used to produce this bile salt. So see, the rest of the heme residue is converted into the bile pigment, biliverdin and bilirubin. So will be defeated, they're defecated through during the defecations. So the color of your feces actually is contributed by this bile pigment. Okay, another role of the liver is storage of vitamins. So the liver is the site of the storage of fat-soluble vitamins A, D, E, K, okay, which are retained in quite large quantities. And the vitamin A is also manufactured in the liver from the plant's pigment carotene. Most of the water-soluble vitamin B complex are retained in smaller quantities. Okay. However, vitamin B12 and the folic acid required for the manufacture of red blood cell in the bone marrow are retained in the liver in substantial amount. So the liver function, they are more on the storage of the fat soluble vitamins. For the storage of minerals, so they are stored at ions, okay, which is breakdown from the hemoglobin, okay, and then to be recycled, okay. Potassium, iron, and many trace elements also can be stored in the liver. Okay, storage of blood, like just now we've mentioned. Okay, liver is the largest internal organ, so they act as a reservoir, the blood reservoir, to store large volume of blood. And then more blood is available to the general blood circulation if the sympathetic nervous system and adrenaline stimulate the vasoconstriction of more blood vessel in the liver. And detoxification is a part of homeostasis. Okay, so in here we just explain briefly. So detoxification, they helps in the liver, that helps to maintain the composition of the blood in a steady state. So the bacteria and other pathogens that enter the bloodstream, okay, the bloodstream via the gut are engulfed by the Kappa cells. Okay, then the drugs, toxin, and poisons taken, okay, taken in with the diet or produced by bacteria somewhere in the body. So when they are in the blood circulation, so they are transported to the liver. So biochemically broken down, okay, so it means by enzymes, so into the harmless substances, okay. So the process are, for example, oxidation, reduction, and so on, okay. So the following the detoxifications, the harmless products are excreted by kidney or eliminated through the defecations. So, however, some toxins, food additives, and drugs can harm the liver. So the liver, they cannot completely detoxify these substances. Okay, for example, long-term excessive intake of alcohol, it causes cirrhosis of the liver, which is the hardening of the liver in some people. Okay, and also can cause what we call the liver cancer. Okay. Food additives can be metabolized into the poisonous or carcinogenic substances by liver activities. Okay. Medicines or drug, paracetamol, if taken in excess, will be changed into the substance which affect the enzymatic reaction in the liver, so resulting the damage of liver and other tissue. And the hormones that release or produce and release by endocrine glands, so they are circulate in the bloodstream to the target tissue or target cells. So once finish the what we call the effect of the hormones, then the hormones also and the excess hormones will be circulated, okay, by the bloodstream to the liver, and these hormones are destroyed in liver. Okay, and also some hormones, they are excreted through kidney in urine. Okay, metabolite conversion, like just now we have mentioned, there is carbohydrate metabolisms, amino acid, the protein, okay, protein metabolisms, and fat metabolisms. But according to our syllabus, we only discuss the carbohydrate metabolism and protein metabolisms. Okay. So these videos, we have described the structure of the liver and also some roles of the liver. Okay. So the metabolisms part will be discussed on the next video. Okay. So for the next video, we will discuss about the carbohydrate metabolisms and also the protein metabolisms in the liver. So that's all for today, and thank you very much. Bye.